Good day, friends, and welcome back to the channel. This is now part 2 of our series where we discuss viruses. Now, we don't talk earlier, say the study of viruses for science, we call them virology. And this one, they important to note. We also don't talk, say, they important to realize, say, for the viruses, then be waiting they call acellular entities. They not be living organisms. They for the middle of both living and non-living things. Now, it's important to note, say, when it comes to viruses, they carry either DNA molecule or they go carry RNA molecule. No virus they carry both, that is both DNA and RNA. No virus they carry such thing together. And this one will make them different from other living organisms because most living cells they carry both DNA and RNA, and both DNA and RNA they perform different functions for living organisms. Now, it is also important to note, say, for viruses they exist for two major phases. They exist either in what they call extracellular phase, or they go exist inside the second one will be intracellular phase. Now, if you say extracellular, that one means say the virus they outside the cell, they within or inside the living cell. We refer to that one as extracellular phase. And if virus dey for inside the extracellular phase, that virus no fit replicate, e no fit reproduce, e no fit make many different copies of itself, and e dey get very few enzymes. The activities wey e dey run dey very little whenever the virus dey for the extracellular phase, meaning say that virus no dey inside any living matter. Now, the second phase wey virus fit exist in na wetin we dey call intracellular phase. This one means say the virus don find im way into a living cell, into a living organism. Now, if the virus find im way into the living organism, we dey talk say that virus dey exist for inside the intracellular phase. Now, if the virus dey for inside the intracellular phase, e don turn a different particle be that. At that time, that virus can't get the capability to divide, to make different copies of itself. E dey very active if e dey inside a living host. This one dey important to note. Now, it also dey important to note as students that viruses, if you come to the cultivation or the culture of viruses, we dey culture viruses for inside the laboratory, culturing them inside living cells. Normally, if we remember our discourse on bacteria, we remember say for bacteria, we dey culture them for inside agar or broth. And all these ones are non-living substances, non-living materials which the bacteria go dey feed on, they grow for inside all those media. But viruses no dey like that. To culture viruses, you fit culture viruses only within a living cell. So many times they culture viruses for inside chick embryo, little chick embryo, chicken embryo, or the embryo of the egg of chickens. We dey inject viruses inside, and we dey culture those viruses inside that environment because na living cell. We'll still do video where we will discuss different ways we will take cultivate viruses. Now, it is also important to realize say viruses, apart from to dey culture them for inside living cells, we dey also purify them based on their weight. E get different processes wey we dey call assays, enzymatic assays wey we dey do to use to purify viruses wey we don culture for inside many different living things. Now, as I don talk before for the other video, you fit check am. Viruses dey fit get envelope or dey fit dey naked, make they no get envelope. So we get enveloped viruses and non-enveloped or naked viruses. Now, e also dey important to note that the viruses we get 5 different stages for the life cycle of viruses. Viruses get life cycle, and that life cycle get 5 different steps or stages. It is important to note this. The first step na the attachment of the virus to the host cell. The virus must attach itself to the host cell if the virus will gain entry into that host cell. The second one na entry into the host cell. First, the virus will attach itself. Then, most of the time, the virus dey shed im capsid, the protein coat wey we don discuss for the first video. The virus go shed am, leave am outside, and then the nucleic material, either na DNA or na RNA, e go inject am into the cell, and that nucleic material go dey inside the host, the new host cell wey this virus wan go dey replicate inside. So you get attachment to the host cell, you get entry into the host cell. Now, if the viral nucleic acid don enter into the host cell, the next thing wey you will get, the next step na synthesis of viral nucleic material for inside the host cell. So within the host cell, the virus go dey replicate, and as e dey replicate, wetin e dey use replicate na the host cell, the materials wey the host cell bin get, the DNA wey the host cell get, the RNA wey the host cell get, the virus go dey use all those materials dey replicate, dey divide into many new viruses. Now, as that one dey happen, we go the fourth step, and the fourth step na assembly, self-assembly of the virus within the host cell. Now, wetin go happen for there na say, as this virus don enter into the host cell, come replicate, come dey plenty for inside the host cell, the virus go come begin dey assemble protein coat. Protein coat wey we don talk for the first video, say protein coat na wetin we dey call capsid. E go come dey, create that capsid around all the different viral nucleic material wey don dey for inside the host cell. Now, the fifth and final step for the life cycle na the release of these virus particles. When this capsid don dey formed for inside the living host cell, e go begin later on, as we don form many of these capsids around these nucleic materials wey don form, we don get many new viral particles. We go come dey release them. That last stage na the release of all these new viral particles back into the environment. So the 5 stages na attachment to the host cell of the virus, entry of the host to the host cell, synthesis of the viral nucleic acid, self-assembly of the virus within the host cell, and the release of virus from the host cell. Now, viruses also, we will end this class by discussing, say, virus get like 3 major things wey been use differ from other living cells. One of them na say viruses na wetin we dey refer to as simple acellular entities. Secondly, viruses dey get either DNA or RNA for their structure. They no dey get both DNA and RNA. Meanwhile, most living organisms get both DNA and RNA, performing different functions within the same cell. Lastly, viruses, they no dey fit replicate outside a living organism. They must colonize or colobi a living organism, dey inside that living organism, before they go fit replicate into 2, 3, or 4. So thank you for listening to this lecture. Make una try to dey share our content, dey like our content, so that our content go dey go wider, we go dey reach many more people, and then we go fit do more of wetin we dey do. Thank you for listening and have a wonderful day.